Hey guys! For today's video, let's talk about proteins. When we hear about protein, we usually associate them into certain kinds of foods such as chicken, eggs, beans, milk, and so on. Well, this is so true. We actually need protein in order to survive. Proteins actually serve as a fuel of our cells. Without these components, without this very important component of certain food, life would be very impossible. So, do you also know that proteins can be also manufactured in the laboratory? Well, scientists have been discovering a lot of different methods in order to produce protein in the laboratory. One of the methods is actually using a bacteria. So, these tiny organisms are really very helpful in order to produce proteins. Scientists engineer these bacteria in which their gene can produce certain types of protein in order to be used in the laboratory or in order to be used in the production of medicine. In addition to that, plasmids can also be inserted into the bacteria and the desired protein can be expressed. This is particularly very important because the scale of production of protein can actually be adjusted. So if we want a lot amount of protein from certain bacteria, we can actually increase the scale and get a lot of proteins from that. So for the production of medicine, a protein from bacteria is really very helpful. However, one of the disadvantages of protein from bacteria is that at certain conditions, proteins cannot be expressed from bacteria. So, the limitation is that there are a few kinds of proteins that bacteria cannot actually produce. So, therefore, scientists have to find an alternative method on how to do that. Okay, so let's look at the other methods. One of the methods is actually to produce the protein from the chemical methods. What is this? So this is actually like a combination of different chemicals in the laboratory in order to produce proteins of interest. This is particularly very important for proteins that are difficult to be expressed by bacteria in the laboratory. And in addition to that, it's very important in the study of post-translational modifications. So post-translational modifications are actually certain modifications that happen inside the cells. So these have been proven to be associated in certain types of diseases such as Alzheimer's disease or the other neurological diseases. So in our body, some of our DNAs or some of our proteins are actually modified. And this modification would lead into certain effects such as diseases and so on. So bacteria couldn't synthesize this post-translationally modified protein. That's why it's very important to synthesize them in the laboratory to study these types of proteins. And one important aspect of life science is also the fluorescent probes. These are very useful in investigating what's happening inside the cells. So some of fluorescent probes are actually made up of proteins, and these proteins can only be synthesized chemically. So that's why this chemically made protein is really very important in the study of biochemistry, biology, or life sciences. Okay, let's move on to the next part. So let's talk about the basic of the protein structure. So a protein is actually consists of different combinations of amino acids. So for example, imagine this one emoji is an amino acid and this other emoji is another amino acid. And if you collect them together, they're going to form a protein. And a long collection of amino acids that are joined together is called a protein, and a shorter fragment of protein is actually called a peptide. If you join two amino acids together, it's going to become a peptide. So it's called dipeptide. And if you have a lot of peptides and you combine them together, it's going to be a protein. Why is peptide very important in the laboratory? Actually, it's very difficult to produce a long protein using chemical method. That's why scientists try to divide these peptides into smaller components, or they try to divide this protein into smaller components in order to easily synthesize it in the laboratory. Then let's proceed into the amino acid. So what is amino acid? Amino acid is an organic molecule that is made up of the basic amino group. So it has amino group here and an acidic carboxyl group and also an organic R group that is unique to each amino acid. So as you have seen here on the 21 different amino acids, all of them have the basic group, amino group, and they also have the acidic carboxyl group, which identifies them as the unique amino acid. In addition to that, they also have different side chains, which further subdivides the categories of the amino acid. So, in these 20 naturally occurring amino acids, they are actually divided into certain categories. So, for example, these three are the positive amino acids, two of them are negative amino acids, and some of them are neutral or uncharged amino acid, and the others are hydrophobic side chains, and also there are special cases of amino acids. If you're going to study about protein chemistry, it's very important to become familiarized or to understand the structures of each of these amino acids because later on this will be very useful in your study. So you need to be familiarized with all of these different kinds of amino acid. Okay, let's move on to peptides. So, as what I have mentioned earlier, in order to synthesize the whole protein, first you're gonna divide it into smaller fragments such as peptides because it's difficult to synthesize longer proteins. Then let's go to the core of this video. Let's learn about the solid phase peptide synthesis. So what is this SPPS? This is actually one of the best used methods in order to synthesize a protein in the laboratory. So there are components in this SPPS method. The first one is the racine, and then followed by a linker, and then the amino acid residue, and the side chain protection group, and the main chain protection group. Okay, let's start with a racine. A racine is actually very important in the synthesis of peptides naturally. So these acts as a support in which your protein or your peptides are attached. So imagine you have this tube for chemical reaction, and inside this tube, you're gonna put all of your reagents, all of your proteins and peptides, and it has a hole below. And then there should be a racine here, and this racine is where your peptide attaches into. So when you add your amino acid into this tube, then it's gonna be attached into the racine, and then the excessive or excess reagents can be washed away without washing your protein, without washing your peptide. So first, a racine, we start with a racine which is followed by a linker. So this linker actually is consist of amino group which will then be used to attach the carboxyl group of the amino acid using the process of condensation. So first, your racine and the linker, and then your first amino acid is going to be attached here, and you get the racine, linker, and the amino acid. Also note that an FMOC is a very common protection group in amino acid because this allows the simultaneous addition of individual amino acid into the next amino acid. So in your next step, you're going to remove that FMOC group. So it's called the FMOC deprotection, which is usually carried out using 20% piperidine in DMF. And after addition of FMOC deprotection reagent, you're going to expose the amino group in this part, which then allows you to add the next amino acid, which has the carboxylic group. And using the condensation reaction, you're going to bind these two amino acids together, and you're going to form a dipeptide. And then your next step again is to remove this FMOC group, which is in the step of FMOC deprotection. And your next step is again the addition of the next amino acid. And the series of FMOC deprotection and the addition of amino acid into your peptide chain actually elongates your peptide and enables you to synthesize a full length or long peptide. And after that, in the last step, you're going to remove this FMOC group at the end of your last peptide. And also, you can remove the protection group of your side chains. As we have mentioned earlier, I showed you the different amino acids, and all of them have different side chains. So, in order to prevent unwanted chemical reactions, side chain protection is very important, and that's why it's added here. So you can remove it at the end of your experiment step. So this solid phase peptide synthesis is very easy and very useful method, and it takes a little bit, it's not so long time. So for example, if you add one amino acid to the next amino acid, it's gonna take, like, the coupling reaction actually takes about 30 minutes to an hour. And then the FMOC protection takes just 5 minutes. But you may want to do it twice because you want to make sure that the FMOC is completely deprotected. And in addition to that, there's actually a test to confirm if your FMOC is present here or it's absent. So you can do different tests that you want. You can try to measure the molecular weight of the amino acid, or you can try the Kaiser test, which, if the FMOC is present, it's going to be white, and if the FMOC is not present, it's going to be blue. So, well, this is just the overview of the solid phase peptide synthesis. So thank you for watching, guys. And if you want me to explain further, please comment down below. And also, please subscribe to my channel if you want to learn more about different stuff about science or biochemistry. I will keep you posted. So thank you very much for watching my video, and I hope you learned something from it. Bye!